At 3 a.m., the world is quieter. The streets are empty, the wind is softer, and thoughts slow down. It's a time when the mind drifts, neither fully awake nor asleep. And in that space, something unexpected begins to grow. Not with leaves or branches, but with threads fine as breath. Fungi. They move quietly, beneath our feet and through forgotten spaces, binding roots, carrying whispers, and weaving life together. Neither plant nor animal, yet close to both, they are the hidden fabric of the living world. So let yourself sink into the silence. Dim the lights, close your eyes if you like, and let your thoughts trail off like spores in a breeze, because at 3 a.m., even the smallest things deserve to be seen. Over 1 billion years ago, the continents were silent stone. No forests stood in the wind, no roots clung to the soil, because there was no true soil to hold. The land was raw, exposed to the sun's harsh ultraviolet light, by day chilled by unbroken darkness at night. Here and there, shallow tidal pools shimmered, carrying traces of early microbial life from the oceans. It was in this stark, alien setting that fungi began their story. Not the mushrooms we think of today, delicate caps and stalks erupting after rain, but thread-like pioneers, weaving through cracks in rock and grains of mineral dust. These ancestral fungi likely resembled simple networks of hyphae, their filaments reaching into microscopic crevices to draw out water, trace minerals, and the remnants of decayed microorganisms washed ashore. They lived in a twilight world between sea and land, where conditions shifted rapidly and survival meant adaptability without motion. For centuries, the deep past of fungi was a mystery, hidden not only by time, but by their fragile anatomy. Soft tissues rarely fossilize. Yet, in the last 2 decades, scientists have uncovered remarkable evidence that rewrites our timeline. Fossilized filaments, branching with the characteristic patterns of modern hyphae, have been found in billion-year-old rocks in Canada's Grassy Bay Formation. Their chemical fingerprints, notably the presence of chitin, a structural component unique to fungal cell walls, match the biology of present-day species. Even older possible traces come from the Arctic's shale deposits, dating to 1.3 billion years ago. These tiny remnants are not proof beyond all doubt, but they strengthen the hypothesis that fungi began their terrestrial presence hundreds of millions of years before plants appeared. In evolutionary terms, fungi may have been the first true colonizers of the land, building the first faint web of life across barren rock. Life on land required more than endurance, it required transformation. Rock had to be broken down, its locked minerals released into forms that could nourish future life. Fungi became masters of this task. Through the secretion of organic acids and specialized enzymes, their filaments could slowly dissolve mineral grains, freeing phosphorus, potassium, and other essential nutrients. In doing so, they created the first fragile protosoils, thin films of organic matter bound to mineral particles. This biochemical weathering not only changed the land's surface, but also altered Earth's atmosphere. By supporting microbial mats and later primitive plants, these early soils helped draw down carbon dioxide, setting in motion feedback loops that would influence global climate for millions of years to come. One of the most intriguing questions in mycology is whether the earliest fungi already formed partnerships with other organisms. Today, Over 90% of plant species form mycorrhizal associations, intimate, mutually beneficial relationships with fungi that exchange water and nutrients for sugars. Some researchers suggest that similar alliances may have existed between ancient fungi and cyanobacteria or algae long before the first true plants evolved. Such partnerships could explain how fungi gained a foothold on land despite its challenges. By working with photosynthetic organisms, they could access a steady supply of organic carbon in exchange for protection and mineral nutrients. In this view, the fungal kingdom's history is inseparable from cooperation, a theme that continues through every chapter of their story. Early Earth's surface was far from stable. Volcanic activity was intense, with frequent eruptions blanketing land in ash and altering local climates. Ice ages came and went, freezing much of the planet for millions of years. Through it all, fungi endured. Their ability to go dormant during inhospitable periods, their microscopic size, and their reliance on chemical rather than photosynthetic energy made them remarkably resilient. Unlike plants, fungi do not require sunlight. Unlike animals, they do not need to seek out food through movement. They persist by weaving into whatever environment they find, breaking it down and reshaping it from within. This quiet persistence is perhaps the key to why they have survived multiple mass extinctions, each time emerging as crucial players in the recovery of ecosystems. Over time, the simple filamentous fungi diversified. Some developed complex fruiting structures to spread their spores more efficiently. Others specialized in decomposing the toughest organic materials, such as lignin in early woody plants. By the late Precambrian and early Paleozoic eras, fungi had become integral to the Earth's biosphere, forming intricate ecological webs that connected microorganisms, plants, and animals in ways still only partly understood. One of the most iconic fossil clues from this period is Prototaxites, a towering, trunk-like organism that lived around 420 million years ago. For decades, scientists debated whether it was a plant, an alga, or a fungus. Today, the evidence strongly supports that Prototaxites was fungal, and at up to 8 meters tall, it would have been one of the largest living things on land at the time. Its existence suggests that fungi were not just silent networks underground, but, at times, dominant above-ground presences in ancient landscapes. By the time the first true plants evolved roots, fungi had already shaped the land into something more hospitable. They had weathered rock into soil, built nutrient cycles, and perhaps even guided plants in their early evolutionary steps. In return, plants would later provide fungi with abundant organic carbon, creating one of the most successful symbiotic partnerships in Earth's history. Without fungi, the colonization of land by plants might have been delayed by tens or even hundreds of millions of years, altering the trajectory of life on Earth. This makes the fungal kingdom not a supporting character, but a central architect in the grand evolutionary narrative. From those ancient, unseen filaments to the vast mycelial networks that now thread through forests and grasslands, fungi carry a legacy older than most forms of life we see around us. Their beginnings are a reminder that the forces shaping our world often work slowly, invisibly, and without seeking recognition. We will descend deeper into the hidden kingdom, exploring its taxonomy, its astonishing diversity, and the many forms fungi take as they continue their billion-year story. In the darkness beneath the forest floor, in the damp crevices of stone, in the air we breathe and the oceans that cover most of our planet, fungi are everywhere. They are not simply scattered. They form a vast interconnected kingdom that operates by its own rules. To understand fungi is to look beyond the occasional mushroom and into a realm of diversity that defies easy categorization. For centuries, scientists struggled to classify fungi. Were they plants? They grew rooted in place. They produced spores. Or were they animals? Their cells contained chitin. That same compound is also found in insect exoskeletons. The truth is that fungi are neither. They belong to their own kingdom, a distinct branch of the tree of life, more closely related to animals than to plants, yet profoundly different from both. Modern molecular studies have confirmed this uniqueness. DNA sequencing has revealed something remarkable. Fungi share a common ancestor with animals that lived more than 1 billion years ago. From that point, they branched off into their own evolutionary line, a lineage that has since exploded into millions of species. Many of them remain undescribed. Today, estimates suggest that around 150,000 species have been formally identified, but the true number could be between 2.2 and 3.8 million. Within this hidden kingdom, diversity unfolds along several evolutionary lines. Chytridiomycota, also called chytrids. They are the simplest and most ancient fungi, often aquatic. Their spores can swim with a single flagellum. Some are decomposers. Others parasitize plants, algae, or even amphibians. Zygomycota, known for their resistant structures called zygospores. This group includes familiar bread molds. They also live in soils, where they help decompose organic matter. Glomeromycota, specialists in forming arbuscular mycorrhiza. They live inside the roots of plants, and they are essential for nutrient exchange. Ascomycota, also called sac fungi, the largest group of all. They produce spores in small sac-like structures. This group includes yeasts, truffles, and many plant pathogens. Basidiomycota, or club fungi, known for producing elaborate fruiting bodies. The classic mushrooms, puffballs, and bracket fungi, as well as rusts and smuts that affect crops. These divisions are not mere academic distinctions. Each one represents a unique evolutionary strategy for survival and reproduction, morphological variety. Fungi take forms so varied that the word mushroom barely scratches the surface. Some exist as unicellular yeasts, invisible without a microscope. Others grow as vast underground mycelial networks, their reach measured in kilometers. Certain species produce luminous fruiting bodies that glow in the night. Others form delicate, almost invisible crusts on tree bark or stone. Their spores, the primary means of reproduction, are just as diverse. Some are launched into the air by microscopic catapults, while others rely on insects, water currents, or even raindrop splashes to carry them to new territory. Spore walls can be smooth, spiny, or intricately patterned, each adaptation shaped by the environments in which they evolved. The taxonomy of fungi is deeply tied to the roles they play in ecosystems. Saprotrophs, decomposers that recycle dead organic matter, breaking down complex molecules into simpler forms that other organisms can use. Mutualists, partners in symbiotic relationships, such as mycorrhizal fungi with plants or lichens. partnerships between fungi and photosynthetic organisms like algae or cyanobacteria, parasites and pathogens, species that draw nutrients from living hosts, sometimes causing disease, but often playing important regulatory roles in populations. Endophytes, fungi that live quietly within plant tissues, often providing protection against herbivores or drought in exchange for shelter and nutrients. Fungi are not limited to soils and forests. They also live in deserts. They survive within Antarctic ice, on the deep sea floor, and even inside animal bodies. Extremophilic fungi can tolerate high salt. They endure high pressure and even intense radiation. Black yeast-like fungi have been found thriving on the walls of the damaged Chernobyl nuclear reactor. They appear to use radiation itself as an energy source. The ocean alone is an underexplored frontier for mycology. Marine fungi inhabit mangroves, salt marshes, and deep-sea sediments, playing roles in decomposing organic matter and forming relationships with marine plants and animals. Despite advances in molecular techniques, fungal taxonomy remains a moving target. New sequencing technologies constantly reveal hidden lineages. They challenge many of the older groupings. Some species once thought to belong to a single genus have now been split into dozens. Others have been merged as their genetic kinship became clear. This fluidity is not a weakness of science. It is a testament to the complexity of the fungal kingdom. Like the mycelium itself, our understanding grows in branching and interwoven patterns. The map reshapes itself as we continue to explore. Understanding fungal diversity is not just an academic exercise. Different groups of fungi play distinct roles in global nutrient cycles, in plant health, and even in climate regulation. A loss in one part of the fungal kingdom can ripple outward. It can affect entire ecosystems. Conversely, the discovery of new fungal species often leads to new medicines, industrial enzymes, or agricultural innovations. We live on a planet where the majority of fungal species remain unnamed, their ecological roles unstudied. The hidden kingdom continues to evolve, adapt, and diversify, much as it has for over 1 billion years. To step into it is to realize that the surface world we see is supported by a vast, unseen foundation. Beneath every step you take lies a world in constant motion, but it moves without haste, without noise, and without revealing itself to the casual glance. To most eyes, soil is simply the ground. To fungi, it is an endless expanse of potential, a space to explore, to connect, to transform. The true body of most fungi is not the mushroom we see above the surface, but the mycelium, an intricate, living architecture hidden from view. Mycelium is composed of hyphae, microscopic filaments that branch, fuse, and weave together into vast, interconnected webs. Each hypha is a tube of living cytoplasm encased in a cell wall made primarily of chitin. At its growing tip, the hypha secretes enzymes that break down surrounding organic matter or minerals, absorbing the resulting nutrients to fuel further growth. Hyphae grow directionally, sensing chemical signals in their environment. When they encounter an obstacle, they branch around it. When they find a rich source of nutrients, they thicken and multiply, creating dense mats of activity. In this way, the mycelium is both exploratory and opportunistic, a living network that adapts to the landscape in real time. The scale of mycelium is difficult to comprehend. In a handful of healthy forest soil, there can be several kilometers of fungal hyphae. In Oregon's Blue Mountains, a single clone of the honey fungus, Armillaria ostoyi, spans over 2,385 acres, making it the largest known organism on Earth by area. Its network is estimated to be thousands of years old, persisting by slowly expanding and digesting dead wood. Such scale is not limited to forests. Grasslands, wetlands, tundra, all harbor mycelial networks that may connect thousands of individual plants and other fungi into systems of shared resources and information. One of the most striking aspects of mycelium is its ability to move resources across long distances. Through processes known as translocation, nutrients absorbed in one part of the network can be transported to another. In mycorrhizal systems, this allows fungi to shuttle water and minerals from areas of abundance to plant roots in drier or poorer soils. But mycelium does more than move nutrients. It also transmits chemical signals. When one plant in a network is attacked by herbivores or infected by a pathogen, the associated fungi can help relay warning signals to neighboring plants. These plants may then preemptively produce defensive compounds, increasing their resilience. While the mechanisms are still being studied, this communication blurs the boundaries between individual organisms and suggests a collective dimension to life underground. Mycelium is not only a passenger in the soil, it is a builder. As hyphae grow, they bind soil particles together, improving structure and stability. This increases water retention and creates microhabitats for bacteria, microfauna, and other fungi. Over time, this structural engineering shapes the porosity and fertility of entire ecosystems. Certain fungi, known as mycorrhizal mutualists, contribute significantly to carbon storage in soils. By depositing organic carbon compounds into stable soil aggregates, they help lock away carbon for decades or centuries, influencing global climate cycles. In forest ecosystems, the intertwining of plant and fungal networks creates what some researchers call the wood wide web. A single fungal individual may connect the roots of different tree species, allowing them to share resources. Seedlings shaded under a dense canopy may receive carbon from mature trees through these fungal bridges, increasing their survival chances. This interconnection challenges the traditional idea of competition as the sole driver of forest dynamics. Cooperation, mediated by fungal networks, emerges as an equally powerful force shaping community structure. The mycelial network is both robust and fragile. Its vastness allows it to bypass local damage. If one section is destroyed, others can compensate. Yet it is sensitive to large-scale disturbances. Soil compaction, pollution, and habitat fragmentation can sever connections and reduce its functionality. Climate change poses additional threats, altering temperature and moisture regimes in ways that may disrupt these ancient underground systems. While terrestrial mycelia are best studied, similar filamentous networks exist in aquatic environments. Freshwater fungi grow within submerged wood, plant matter, and sediments, forming hidden webs that cycle nutrients and decompose organic debris. Even in deep-sea sediments, fungal hyphae have been observed intertwining with bacterial colonies, suggesting that the architecture of mycelial life is as ancient and universal as the kingdom itself. Above ground, mushrooms come and go with the seasons. Below ground, mycelium is always at work, a patient, unending presence that shapes the living world. Without it, soils would erode, nutrient cycles would collapse, and many plants would fail to thrive. Mycelium is not just the hidden body of fungi, it is one of the central infrastructures of life on Earth. Beneath the surface of every healthy forest, grassland, or garden, there is a quiet exchange taking place, a transaction that began hundreds of millions of years ago and has never ceased. It is not a market of money, but of molecules, carbon for nutrients, water for shelter, survival for survival. This is the world of Mycorrhiza, one of the most successful and enduring partnerships in the history of life on land. The story of Mycorrhiza begins more than 450 million years ago, when the first plants attempted to colonize barren continents. Without deep roots, these early pioneers could not reach water far below the surface or access many essential minerals locked in rock. Fungi, already well adapted to terrestrial life, could. Their thin, exploratory hyphae penetrated cracks in stone, dissolving minerals and drawing moisture from tiny soil pores. Somewhere in this shared landscape, a bargain was struck. Plants would supply fungi with carbon in the form of sugars produced through photosynthesis, and fungi would deliver water and nutrients in return. This agreement was not sealed by intention, but by evolution. Those plants and fungi that cooperated survived, reproduced, and passed on their traits. Over time, the partnership became not the exception, but the rule. Today, over 90% of all plant species maintain mycorrhizal relationships, a testament to the effectiveness of this alliance. While the underlying principle is universal, exchange, the architecture of mycorrhizal relationships takes different forms. Arbuscular mycorrhiza, the most ancient and widespread type, formed primarily by glomeromycota fungi. Hyphae penetrate plant root cells, forming tree-like structures called arbuscules where nutrient exchange occurs. Fungi are critical for crops like wheat, maize, and rice. Ectomycorrhiza, found in many forest trees such as oaks, pines, and birches. Hyphae wrap around root tips, forming a sheath and extending into the surrounding soil, but do not enter the root cells themselves. Fungi include many familiar mushrooms like boletes and chanterelles. Ericoid mycorrhiza, specialized for plants in nutrient-poor, acidic soils, such as heaths and blueberries. These fungi help their hosts access nitrogen bound in complex organic matter. Orchid mycorrhiza. Orchids depend on fungal partners to germinate and grow, as their tiny seeds contain almost no stored nutrients. In some species, this dependence lasts throughout their lives. The exchange in mycorrhizal networks is not random, it is finely tuned. Fungi deliver phosphorus, nitrogen, and trace elements directly to plant roots, often in forms that plants could not obtain on their own. In return, plants provide fungi with carbohydrates, sometimes up to 20% of the sugars they produce. This flow is dynamic. If a plant is shaded and produces less sugar, the fungi may reduce nutrient delivery to it, directing resources toward more productive partners. Similarly, plants can allocate carbon preferentially to fungi that provide the most nutrients, creating a balance of trade that benefits both sides. In ecosystems with multiple plant species, mycorrhizal fungi often connect them into shared networks. Through these networks, carbon can move from one plant to another, water can be redistributed, and chemical signals can travel. This connectivity has surprising ecological consequences. Seedlings growing in deep shade may receive carbon subsidies from sunlit neighbors. Stressed plants may receive drought relief from more resilient ones. Research by forest ecologist Suzanne Simard has shown that some large, old trees, sometimes called mother trees, use mycorrhizal networks to support the growth of younger plants, even those of different species. In this way, Mycorrhiza shapes the structure and diversity of entire plant communities. Mycorrhizal fungi do more than feed plants. They help protect them. Some fungi produce compounds that deter root pathogens or herbivorous insects. Others trigger a plant's own defense mechanisms, priming it to respond faster and stronger to attack. In nutrient-poor or drought-prone soils, mycorrhizal plants often show higher survival and reproduction rates than those without fungal partners. While mycorrhiza is most commonly studied in terrestrial ecosystems, similar partnerships occur in aquatic environments. Certain aquatic plants, such as seagrasses, form associations with fungi that function much like their terrestrial counterparts, aiding in nutrient uptake from sediments. Despite its resilience over geological time, mycorrhiza is vulnerable to human disturbance. Intensive agriculture, deforestation, pollution, and soil compaction can disrupt fungal networks, reducing their effectiveness or eliminating them altogether. Excessive use of chemical fertilizers can make plants less reliant on fungi, weakening the partnership. Climate change, by altering temperature and moisture regimes, may also shift the distribution and functioning of mycorrhizal species. Loss of mycorrhiza has cascading effects. Without these networks, plants are less able to cope with stress. Soils lose stability, and carbon storage declines. The collapse of this underground infrastructure can destabilize entire ecosystems. Restoring and maintaining mycorrhizal networks is becoming a key strategy in ecological restoration and sustainable agriculture. Farmers inoculate crops with beneficial fungi to improve yields and reduce fertilizer use. Conservationists protect old-growth forests not just for their trees, but for the ancient fungal networks beneath them. In the face of global change, the survival of many plant species and the ecosystems they anchor may depend on this partnership that began nearly 500 million years ago. Mycorrhiza is not simply an interaction, it is a foundational process that makes terrestrial life possible. When a leaf falls in the forest, its story does not end. It changes authors. Sunlight, once trapped in its green flesh, begins its slow release. Fibers woven by the tree's growth loosen, sugars bleed into the soil, and the scent of life's return to the earth rises in the air. At the heart of this transformation are the fungi, quiet chemists and master architects of decay. Decomposition is more than rot, it is a recycling of existence. Without decomposers, the dead would accumulate, nutrients would remain locked in unusable forms, and ecosystems would grind to a halt. Fungi are among the most efficient decomposers on the planet. Their enzymes can break down materials that resist most other forms of decay, from the cellulose in plant cell walls to the lignin that makes wood rigid. While bacteria also play vital roles in decomposition, fungi excel at processing large, complex molecules. This is due to their filamentous growth form, which allows them to penetrate deeply into organic matter and secrete enzymes directly into their substrate. Different fungi specialize in different kinds of decay. White rot fungi, mostly Basidiomycota, can fully break down lignin, leaving behind a pale fibrous residue. They are the only organisms known to completely degrade this tough polymer. Brown rot fungi target cellulose and hemicellulose while modifying, but not fully consuming, lignin. This leaves behind a crumbly brown residue rich in lignin, which contributes to soil formation. Soft rot fungi, often Ascomycota, attack cellulose in wood under damp conditions, creating cavities in the cell walls. These fungi release an array of oxidative and hydrolytic enzymes, lacases, peroxidases, cellulases, capable of dismantling plant tissues, molecule by molecule. Through decomposition, fungi return carbon, nitrogen, phosphorus, and other elements to the ecosystem. Carbon that was fixed from the atmosphere by photosynthesis eventually re-enters the air as CO2 through fungal respiration, continuing the carbon cycle. Nutrients like nitrogen and phosphorus, once trapped in dead matter, become available to plants and other organisms, fueling new growth. In forests, fallen wood and leaf litter represent massive nutrient reservoirs. Without fungi, these stores would remain inaccessible, effectively locking away resources essential for life. The role of fungi in decomposition also shapes ecological succession. In newly disturbed environments, volcanic landscapes, landslides, or abandoned agricultural fields, fungi are often among the first colonizers. By breaking down pioneer plant litter, they enrich the soil, enabling more complex plant communities to establish. Over decades and centuries, this builds deep, fertile soils capable of supporting diverse ecosystems. Not all decomposers work at the same pace or in the same materials. Some fungi specialize in leaf litter, others in dead wood, animal remains, or even insect exoskeletons. Certain species decompose keratin, the tough protein in hair and feathers. Others feed on chitin, the material that makes up insect shells and fungal cell walls themselves. Some fungi occupy highly specific niches, such as dung fungi, whose spores germinate only after passing through the digestive system of herbivores. These species play crucial roles in breaking down waste and returning nutrients to the soil. Decomposition is often a collaborative process. Fungi frequently work alongside bacteria, invertebrates, and other organisms to break down complex materials. Termites, for example, cultivate fungi in their nests to help digest wood, while certain beetles carry fungal spores to fresh logs, inoculating them for decomposition. In aquatic environments, fungi contribute significantly to breaking down fallen leaves, algae, and dead organisms, maintaining nutrient flow in rivers, lakes, and coastal zones. The activity of decomposer fungi has direct implications for the global climate. By controlling the rate at which carbon is released from organic matter, fungi influence atmospheric CO2 levels. In cold or waterlogged environments, decomposition slows, leading to the buildup of peat, a significant carbon store. But in warmer or drier climates, fungal activity can accelerate, releasing carbon more quickly. Climate change, in turn, affects decomposition rates. Rising temperatures may speed up decay in some ecosystems, potentially releasing more greenhouse gases and creating feedback loops that further warm the planet. Decomposition is not destruction, it is transformation. What appears to us as decay is, for fungi, the creation of opportunity, the conversion of the dead into a foundation for the living. In the web of life, they ensure that nothing is wasted, that the end of one story is the beginning of another. Long before humans wrote their first words, they foraged for food in forests and fields. Among the roots, beneath the leaves, on fallen logs, and in hidden glades, they found treasures unlike anything else: mushrooms. They were not seeds to be planted, nor meat to be hunted, nor grain to be stored. They appeared suddenly, often after rain, as if conjured from the earth itself. Edible fungi have nourished, healed, and inspired humanity for tens of thousands of years, and in doing so, have shaped culture as much as cuisine. Archaeological evidence suggests that humans have eaten fungi since the earliest days of our species. While mushrooms leave few traces in the archaeological record, indirect evidence, such as depictions in ancient rock art and remnants of fungal DNA in prehistoric human remains, points to a long history of mushroom consumption. The discovery of Ötzi, the Iceman, a 5,300-year-old mummy found in the Alps, provided striking confirmation. Among his belongings were two types of fungi: birch polypore, likely carried for medicinal purposes, and tinder fungus, used for starting fires. His journey through the high mountains was accompanied not only by tools and weapons, but by the quiet companionship of fungi. Fungi occupied a place of honor in many early societies. In ancient China, records of medicinal mushrooms date back more than 2,000 years. The revered reishi, called the mushroom of immortality, was believed to promote longevity and spiritual power. In classical Greece and Rome, mushrooms were valued both as delicacies and as symbols of luxury. The Roman Emperor Claudius was famously killed by a dish of poisoned mushrooms. Mesoamerican cultures, including the Aztecs and Maya, used psychoactive mushrooms in religious ceremonies, calling them flesh of the gods. These rituals were intended not for indulgence but for communion with the divine, guided by shamans who understood the fungi's powers. Edible fungi come in extraordinary variety, each with its own flavor, texture, and culinary tradition. Boletes are prized in European cooking for their nutty, rich taste. Chanterelles, golden and trumpet-shaped, carry a delicate fruity aroma. Morels, with their honeycombed caps, are sought after in spring for their earthy depth of flavor. Shiitake, cultivated for centuries in East Asia, are celebrated for their deep umami character. Oyster mushrooms are versatile and easily grown, offering a mild and savory taste, and truffles, hidden underground, release such an intense aroma that they have become one of the most expensive foods in the world. In each case, the mushroom is only the fruiting body, the brief reproductive phase of a much larger fungal organism hidden from view. For much of history, mushrooms were gathered from the wild. Cultivation began in China more than 1,000 years ago with shiitake, followed by methods for growing enoki and wood ear mushrooms. In 17th century France, farmers discovered that the cool, stable conditions of abandoned quarries made ideal environments for cultivating the common button mushroom. Today, mushroom farming is a global industry. Controlled environments allow year-round production, while selective breeding has improved yields and flavors. New techniques, such as substrate sterilization and liquid culture, have made cultivation accessible to small-scale growers as well as industrial operations. Edible fungi are notable for their combination of low calories and high nutrient density. They provide protein, dietary fiber, vitamins, especially B vitamins and vitamin D when exposed to sunlight, and essential minerals like selenium and potassium. They also contain bioactive compounds with antioxidant, anti-inflammatory, and immune-modulating properties. Unlike plants, mushrooms produce ergosterol, which converts to vitamin D2 under ultraviolet light, making them one of the few non-animal dietary sources of this vitamin. Mushrooms have often symbolized transformation, mystery, and fleeting beauty. Their sudden appearance after rain made them seem magical. Their ability to nourish, heal, or poison lent them an air of power. In literature and folklore, mushrooms have been associated with fairy rings, portals to other realms, and encounters with the supernatural. In Japanese haiku, the appearance of specific mushrooms marks seasonal change. In Italian villages, foraging is a cherished tradition passed down through generations. In Russia, mushroom hunting is a cultural event, blending outdoor recreation with culinary reward. The demand for edible fungi has grown dramatically in recent decades. Truffles command thousands of dollars per kilogram in international markets. Specialty mushrooms, once confined to regional cuisines like mai take, lion's mane, and king oyster, are now widely available in supermarkets and farmers' markets. Plant-based diets and interest in functional foods have boosted mushroom consumption. Mycoprotein, a fungal-derived protein source, has become a sustainable alternative to meat. Gourmet restaurants experiment with mushroom-based broths, foams, and fermented products. For all their benefits, mushrooms demand respect. Many edible species have toxic lookalikes, and misidentification can lead to severe illness or death. Traditional knowledge among foragers, knowing which species are safe, when to harvest them, and how to prepare them, is as important as the harvest itself. Edible fungi are more than a source of nourishment. They are threads of connection between human culture and the natural world, between past and present, between the visible and the hidden. The meals they inspire are acts of participation in a relationship that stretches back into prehistory. In the quiet after rain, a flush of mushrooms can transform the forest floor into a gallery of shapes and colors. Scarlet caps shine like jewels against the moss. Delicate veils shimmer in the undergrowth. Ghostly white forms stand among fallen leaves. Their beauty is real, but for some, it hides a lethal chemistry. To step into this world without knowledge is to walk a path where the line between food and poison is perilously thin. Not all toxic fungi are created equal. Some cause only mild digestive upset. Others can destroy organs, damage the nervous system, or even bring about death. The severity of poisoning depends on the species, the amount consumed, and the susceptibility of the individual. Among the most feared are the amatoxin-producing species, such as the deathcap and the destroying angels. Their toxins block protein synthesis in cells and lead to liver and kidney failure. Even a small portion of a single cap can be fatal. Other groups present different threats. Gyrometrine toxins and false morels can cause seizures and liver damage. Arelanine, found in some Cortinaria species, can lead to delayed kidney failure. Muscarine, present in certain inocybe and clitocybe species, overstimulates the parasympathetic nervous system. This causes excessive salivation, sweating, and potentially dangerous changes in heart rate. Why produce such potent toxins? For fungi, it is a form of defense. Fruit bodies are the reproductive structures of a much larger organism, the mycelium, and producing them requires considerable energy. By deterring animals from eating them, toxins help ensure spores can be released and dispersed. Some toxins may also protect against microbial attack, preventing bacteria and other fungi from colonizing the fruiting body before it matures. Many poisonous mushrooms resemble edible species. The deathcap can be mistaken for the paddy straw mushroom, especially when young. False morels mimic true morels. Some toxic boletes differ only subtly in color or spore print from their edible relatives. Such similarities make experience and careful identification essential for safe foraging. In many cultures, this knowledge has been passed down through generations, sometimes reinforced by painful or even fatal mistakes. Not all dangerous fungi kill. Some alter perception in ways that have fascinated and frightened humans for millennia. The most famous are the psilocybin-containing mushrooms, often called magic mushrooms. Species like Psilocybe cubensis and Psilocybe semilanceata produce compounds that interact with serotonin receptors in the brain. They induce altered states of consciousness, vivid hallucinations, and shifts in the perception of time. Archaeological evidence and ethnographic records show that psychoactive mushrooms have been used in spiritual and shamanic practices from Mesoamerica to Siberia. In the rituals of the Mazatec people of Oaxaca, psilocybin mushrooms, teonanacatl, the flesh of the gods, are consumed to facilitate healing and divination. In northern Eurasia, the fly agaric, with its iconic red cap and white spots, holds a prominent place in folklore and ritual. Its psychoactive compounds, muscimol and ibotenic acid, produce effects quite different from psilocybin. Yet shamans in Siberia have long used it to enter trance states. In recent years, research into psilocybin has expanded rapidly. Clinical trials suggest it may help treat depression, anxiety, and addiction when administered in controlled settings. The compound appears to promote neuroplasticity, the brain's ability to form new connections and to reduce activity in brain networks associated with rigid patterns of thought. However, outside of clinical environments, hallucinogenic fungi carry risks. Reactions can be unpredictable. Some people may experience overwhelming anxiety, confusion, or dangerous behavior during intoxication, and there is always the risk of misidentification, mistaking a toxic species for a psychoactive one. Toxic and hallucinogenic mushrooms have shaped myths and warnings across the world. In European fairy tales, the fly agaric marks the spot of enchantment or danger. In some Asian traditions, poisonous fungi are seen as manifestations of malevolent spirits. The phrase, poisonous as a toadstool, has even entered language as a metaphor for hidden danger. In contrast, cultures that use psychoactive mushrooms ritually often regard them as sacred, not a threat, but a doorway. The difference lies as much in context and knowledge as in chemistry. The existence of toxic and hallucinogenic fungi serves as a reminder that the fungal kingdom operates on its own terms. Its gifts are generous: food, medicine, ecological stability, but never without boundaries. Respect, careful study, and cultural wisdom are essential to navigating this landscape safely. From the same kingdom that yields poisons capable of shutting down organs and compounds that alter consciousness come some of humanity's most important medicines. Fungi do not act with intent. They simply produce molecules to survive, to defend, to compete. But in those molecules, humans have found tools to save lives, fight disease, and reshape industries. In 1928, Alexander Fleming returned from a holiday to find that a mold, later identified as Penicillium notatum, had contaminated one of his bacterial cultures. Around the mold, the bacteria had been destroyed. Fleming had discovered penicillin, the first widely effective antibiotic. This breakthrough marked the beginning of the antibiotic era, transforming medicine. Infections that had once been deadly became treatable. During World War II, mass production of penicillin saved countless soldiers' lives. It also sparked a surge in the search for other fungal metabolites with pharmaceutical potential. Fungi produce a staggering diversity of secondary metabolites. These are chemicals not essential for their immediate survival, but often crucial in competition with other organisms. Many of these compounds have found applications in medicine. Immunosuppressants. Cyclosporin, produced by Telepocladium inflatum, revolutionized organ transplantation by preventing rejection. Cholesterol-lowering agents. Statins, such as lovastatin from Aspergillus terreus, reduce cholesterol synthesis and lower the risk of cardiovascular disease. Anti-cancer agents. Compounds like lentinin from shiitake mushrooms have shown immune-boosting and anti-tumor properties. Antifungals. Griseofulvin, derived from Penicillium species, treats fungal infections in humans and animals. Long before the era of antibiotics, fungi already played a role in traditional healing systems. In China, Ganoderma lucidum, the reishi mushroom, was valued for its supposed longevity benefits. Cordyceps species were used to boost stamina and vitality. In Russia and Eastern Europe, Inonotus obliquus, known as chaga, was brewed into teas for its anti-inflammatory properties. Modern research has begun to validate some of these uses. Scientists have identified bioactive compounds with antioxidant, antiviral, and immune-modulating effects, but not all traditional claims withstand scientific scrutiny. Rigorous clinical trials are essential to separate potential therapies from folklore. Fungi's utility extends beyond medicine. Mycelium can be cultivated into lightweight, durable materials for packaging, insulation, and even building components. Unlike plastics, these mycomaterials are biodegradable and can be grown from agricultural waste. In biotechnology, fungal enzymes are harnessed to produce biofuels, degrade pollutants, and manufacture food products. Enzymes from Aspergillus and Trichoderma species are used in brewing, baking, and textile processing. Lacases from white rot fungi can even break down dyes and other pollutants in wastewater treatment. Research into fungal metabolites is expanding with advances in genomics and metabolomics. Many fungi harbor so-called silent gene clusters, DNA sequences that code for potentially useful compounds but are not expressed under standard laboratory conditions. By activating these clusters, scientists hope to discover entirely new classes of drugs. The rise of antimicrobial resistance has renewed urgency in the search for novel antibiotics. Once again, fungi are prime candidates. Endophytic fungi, Those living inside plants without causing disease are proving to be rich sources of unique chemical structures with antimicrobial potential. Yet challenges remain. Overharvesting of wild medicinal fungi can threaten ecosystems. Cultivation methods must balance efficiency with environmental impact. One solution comes from synthetic biology, transferring fungal genes into easily cultured microorganisms to produce compounds without depleting wild populations. The journey from a fungal metabolite to a life-saving drug is long and complex. It requires not only discovery and isolation, but also careful testing for safety and efficacy, large-scale production, and distribution. Each success deepens the paradox of the fungal kingdom. The same biology that can kill can also cure. The health of an ecosystem is written not only in its trees and animals, but in its quiet foundations, the soil, the roots, the networks of life that remain hidden. In those foundations, fungi are not merely inhabitants, they are architects. They shape structure, guide flow, and design the invisible scaffolding upon which entire communities stand. In forests, fungi are embedded in nearly every process. Mycorrhizal networks link trees together, balancing competition and cooperation. Saprotrophic fungi recycle fallen wood and leaves, releasing nutrients to sustain new growth. Pathogenic fungi, though often seen as destructive, regulate populations, preventing any one species from dominating. The distribution of fungi in a forest is not random. Certain species form tight relationships with specific trees, Boletus edulis with pines, Amanita muscaria with birches, influencing the composition of plant communities. These associations can determine which seedlings survive and which fail, effectively shaping the next generation of the forest. In tropical rainforests, fungal diversity reaches staggering levels. Endophytic fungi live inside leaves and stems, defending their hosts from herbivores and pathogens. Leaf litter fungi decompose organic matter almost as quickly as it falls, driving one of the fastest nutrient cycles on Earth. In grasslands where trees are sparse and root systems dominate, fungi still play critical roles. Arbuscular, mycorrhizal fungi enhance the drought tolerance of grasses, increasing resilience in the face of seasonal extremes. Saprotrophic fungi recycle dead plant material, contributing to the formation of rich, dark prairie soils. Fungi also influence grassland stability. By binding soil particles, mycelium reduces erosion from wind and water. In heavily grazed systems, dung fungi help break down waste, returning nutrients to the soil and supporting invertebrate communities. Grasslands that lose their fungal diversity often show declines in productivity and biodiversity as the delicate balance of nutrient cycling collapses. Restoration efforts increasingly focus on reintroducing native mycorrhizal fungi to revive degraded pastures. Wetlands, marshes, bogs, swamps are some of the most carbon-rich ecosystems on Earth. Fungi here face unique challenges: waterlogged soils, low oxygen levels, and high organic matter content. Specialized species thrive in these conditions, breaking down plant litter slowly, allowing peat to accumulate over centuries. In bogs, certain fungi form associations with mosses like sphagnum, enhancing their ability to capture nutrients in nutrient-poor water. In mangroves, fungi colonize submerged roots and decaying wood, recycling organic matter in tidal rhythms. Marine fungi remain among the least studied components of ocean ecosystems, yet their influence is profound. They inhabit nearly every marine habitat. Surface waters, deep-sea sediments, coral reefs, and the microbiomes of marine animals. Some marine fungi decompose driftwood and seaweed, releasing nutrients that feed plankton, the base of oceanic food webs. Others form symbioses with seagrasses or sponges, contributing to their hosts' health and resilience. In coral reefs, certain fungi may play roles in nutrient cycling and the breakdown of organic debris, though these interactions are only beginning to be understood. Deep-sea fungi, adapted to high pressure and low temperature, are particularly intriguing. Their metabolic pathways may hold keys to novel enzymes and bioactive compounds with industrial and medical applications. In every ecosystem, fungi act as keystone players, species or groups whose impact is disproportionately large relative to their abundance. The removal of key fungal partners can trigger cascading effects: tree decline in forests, loss of soil fertility in grasslands, reduced carbon storage in wetlands. Similarly, shifts in fungal communities can signal environmental change. Invasive fungal pathogens, such as those causing white nose syndrome in bats or chytridiomycosis in amphibians, can destabilize entire ecosystems. Fungi influence global climate through their roles in carbon and nutrient cycles. In forests, mycorrhizal fungi enhance carbon sequestration by increasing plant productivity and storing organic matter in soils. In wetlands, slow fungal decomposition rates allow carbon to accumulate as peat. In oceans, marine fungi contribute to the biological pump, transferring carbon from surface waters to the deep sea. Climate change, in turn, alters fungal communities. Rising temperatures, shifting rainfall patterns, and ocean acidification all affect fungal diversity and function. Some species may expand their ranges, while others decline, with unpredictable consequences for ecosystem stability. Though invisible to most, fungi design the flow of energy and matter in ecosystems. They determine who thrives and who fades, not through dominance, but through the quiet shaping of relationships and resources. Without their architecture, the visible structures of life, forests, grasslands, reefs, would collapse. The story of fungi is one of resilience. They have endured ice ages and mass extinctions, volcanic winters and shifting continents. They have adapted to heat and cold, drought and flood. Yet the pace of change now unfolding is unlike anything in their billion-year history. The question is no longer whether fungi can adapt, but how, and whether they can do so in time to maintain the delicate balances they sustain. Climate has always shaped fungal communities. Temperature, moisture, and seasonal rhythms determine which species can survive and how they function. A shift in any of these factors alters the composition of fungal assemblages, changes that ripple through the plants, animals, and microbes connected to them. Rising global temperatures are already influencing fungal phenology. In temperate regions, mushroom fruiting seasons have lengthened, with some species producing fruit bodies earlier in spring and later into autumn. While this might seem beneficial, the shifts can disrupt ecological synchrony, affecting the species that depend on those fungi for food or symbiosis. As climate zones move, so do fungi. Species adapted to cooler conditions may retreat to higher altitudes or latitudes, while heat-tolerant fungi expand into new areas. This redistribution can lead to novel ecosystems, combinations of species that have never coexisted before. Such changes can be double-edged. New mycorrhizal associations might emerge, potentially benefiting plants in stressed environments. But the arrival of non-native fungi can also disrupt existing networks, outcompete local species, and alter nutrient cycles. Moisture is one of the most critical factors for fungal activity. Extended droughts reduce the growth of mycelium and the formation of fruit bodies, diminishing both decomposition and nutrient exchange with plants. In forests, reduced mycorrhizal activity during drought can limit tree growth, increasing vulnerability to pests and disease. Conversely, intense rainfall and flooding can favor certain opportunistic fungi while washing away spores and damaging delicate soil structures. In wetlands, changes in water levels can accelerate decomposition, releasing stored carbon and undermining the long-term carbon sink function of peatlands. Climate change is also shifting the ranges of fungal pathogens. Warmer winters allow some plant and animal diseases to persist year-round or spread into new regions. Crops face increasing threats from rusts, smuts, and mildews, while wildlife suffers from emerging diseases such as chytridiomycosis in amphibians and white nose syndrome in bats. In human health, the rise of Candida auris, a multi-drug-resistant yeast, has been linked in part to warming temperatures that may have selected for strains able to survive at mammalian body temperatures. Fungi are major players in the global carbon cycle. As decomposers, they control how quickly carbon stored in organic matter returns to the atmosphere. Warming can accelerate decomposition in some ecosystems, increasing carbon dioxide emissions. In others, drought may slow fungal activity, reducing carbon release, but also limiting nutrient availability for plants. The balance of these processes will influence whether ecosystems act as carbon sinks or sources in the coming decades. Mycorrhizal fungi add further complexity. Some enhance carbon storage by promoting plant growth and stabilizing organic matter in soils, while others may accelerate decomposition under certain conditions. Fungi possess remarkable adaptive capacities. Their vast genetic diversity, rapid reproduction, and ability to form dormant spores give them tools to survive environmental extremes. Some species can alter their physiology to tolerate higher temperatures or fluctuating moisture levels. Others may shift to different hosts or substrates as conditions change. Human actions can support this resilience, protecting diverse habitats, reducing soil disturbance, and maintaining healthy plant-fungal networks increase the chances that fungi will adapt successfully. In agriculture, fostering beneficial fungi through reduced tillage, organic amendments, and mycorrhizal inoculation can help crops withstand climate stress. Our understanding of fungi in a changing climate is still in its early stages. They are less visible than plants and animals, and their global diversity remains largely undocumented. Without baseline data, it is difficult to track changes or predict future trends. Investing in fungal research is not merely an academic pursuit. It is an ecological necessity. From food security to carbon sequestration, disease control to habitat restoration, fungi will play pivotal roles in how ecosystems respond to the challenges ahead. The kingdom of fungi is not silent, But it is subtle. It moves slowly, almost invisibly, beneath our feet and across the hidden corners of oceans and forests. It has never been static, never still. Today, though, its designs are being reshaped, not only by time and chance, but by us. In the quiet spaces beneath our feet, and in the unseen threads of soils, wetlands, and reefs, the architects of ecosystems are already adjusting their designs. The future they are building will be shared by all. So the next time you see a mushroom on a forest path, in the grass after rain, or rising quietly from a fallen log, pause for a moment. Remember that you are looking at the briefest sign of a vast and ancient kingdom, a kingdom that recycles death into life, that connects trees into communities, that breathes through the earth itself. It has been here long before us, and if we choose wisely, it will remain long after.